Hey, welcome back. Um, when we ended our last lecture video, I ended with this slide, and I emphasized that with this course understanding and relationships between these major groups of green plants, or the Virida plantae, that includes the green algae, our non-vascular plants, or bryophytes, and our vascular plants, that it's difficult for us to reconcile and understand the evolution of different morphological characters that gave rise to that transition onto land, as well as the transition to vascular tissue. And I emphasize that until we understand the evolutionary relationships better, it almost seems miraculous that we have these major transitions between these very different groups of organisms. But as we'll see, as we break these down into as we get into the details of many of these groups, and we understand better the evolutionary relationships, the evolution of these morphological characteristics becomes much more clear. And so right now, depicted on this, I have 3 synapomorphies shown for land plants. We'll see that this is actually going to expand a little bit as we understand these relationships better. But one of those is obviously the transition to land. One is the evolution of having an embryo associated, and then having multicellular reproductive structures. And I'll build on all of these characteristics as we continue on through this lecture. Well, the way I have this is really coarse. And it turns out that green algae are not just one lineage and represent multiple different lineages. And so if we take a look at that, there's really 2 major groups of green algae. The first of those is the Chlorophyta, and the second is the Charophyta. And although these were morphologically similar to each other, it turns out that they have very different characteristics. And so I just want to highlight a couple of those. So Chlorophyta is made up of about 4,300 species. And these species are primarily, these species are found in both freshwater and marine environments. So marine being saltwater environments. They are made up of a couple of species that are of note and worth mentioning. The model species Chlamydomonas. Chlamydomonas has been used as a model for understanding the evolution of the chloroplast as well as photosynthesis. So that's a model system that's been used. That's the Chlorophyta. Charophyta, on the other hand, has about the same number of species, around 4,500 species, but these are restricted to freshwater environments. And importantly, these are typically found in nearshore freshwater environments. And I'll just highlight the genus Chara. The name Charophyta comes from that group. These represent actually 2 major groups of green algae. And when they have some important differences in them. And one of those differences that I want to highlight is actually a difference in the cell division in these 2 groups. So in the Charophycean green algae, or the chlorophytes, they divide, when their cells are dividing in mitosis, they divide in using something called the phycoplast. And so what a phycoplast is when the nuclei are actually separated by equatorial microtubules. And so that's shown here, where we see 2 nuclei dividing. So these are separating from each other, and the phycoplast is forming with those equatorial, that meaning in across the equator of the cell. We see these equatorially arranged microtubules. And this is all from ultrastructural studies looking at how cell division is initiated in these 2 different groups. When we look at charophytes, or the Charophycean green algae, these actually undergo something that we call phragmoplastic, or a phragmoplast is involved in the cell division. And the reason that I'm telling you this will become clear in just a couple of slides. And so what the phragmoplast is that actually the nuclei are separated by a cell plate. The nuclei separated by a cell plate. And so that phragmoplast is shown here, the cell plate initiating, and we notice that the microtubules are actually arranged in a different orientation than we see in the chlorophytes. So this is an ultrastructural thing. These details aren't incredibly important, but what is important is that there's the main difference between the chlorophytes and the charophytes. And this is down to the level of something as important as cell division happening in two quite different ways. And so if we just take a quick look at how cell division in charophytes happens, I just want to note that this phragmoplastic, this is a word that you should use at the next cocktail party that you attend, phragmoplastic. I realize that my jokes land on deaf ears here, but I can try. That phragmoplastic cell division happens in charophytes, and it also happens in land plants. And that's really the key here. In both land plants and in charophytes, we see the same type of cell division where the spindle fibers persist, small vacuoles aggregate between the nuclei. This is actually the phragmoplast, or that cell plate forming, and then that cell plate forms in the phragmoplast between those two nuclei. And really, the reason I'm emphasizing this phragmoplastic cell division is because this is something that's shared with land plants. And as molecular data, or DNA sequence data, has been used more and more for reconstructing phylogenetic relationships, what actually was discovered with some of the early molecular phylogenetic studies is that green algae, as we talked about them, including both Chlorophycean and Charophycean green algae, are paraphyletic. These are not a monophyletic group. And so really, what we're seeing is that the chlorophytes and the charophytes, on this slide, I have charophytes represented by two lineages, Charales and Coleochaetae. Don't worry about that too much. These are the charophytes. What we see here is that these two are not monophyletic. They actually represent two separate lineages of green algae. And that's important because once the molecular phylogenetic results showed this, then we can start to look at all of the different characteristics that are different between chlorophytes and charophytes. And we see that charophytes actually share a huge number of morphological, biochemical, and molecular evidence that unite them with land plants. And so it makes it a little easier to understand the evolution of many of these characters that we see in land plants that we don't see in some of the green algal relatives. And so I've listed some of those here. So these are all synapomorphies for the clade that contains both land plants and the Charophycean green algae. And that, of course, includes the phragmoplast. So we have phragmoplast formed during cell division. But there are a number of other synapomorphies that are shared between these two groups as well, including asymmetric sperm flagellae, filamentous growth, a glycolate oxidase enzyme system, and the retention of the egg and the zygote on the plant. And many of these are actually thought to be pre-adaptations to the movement from an aquatic environment where green algae are all strictly aquatic, including charophytes, to land. Moving on to land presents a bunch of challenges. And so I'll come back to a couple of these and how they're pre-adaptations, but just to give you a little bit of a heads up, this glycolate oxidase and filamentous growth are two that are really advantageous once you're moving towards a non-aquatic lifestyle. But I will come back to those. The last piece that I put on here is one molecular evidence. TUF-A is a chloroplast gene, a gene that's found in the chloroplast genome, and that's been moved to the nucleus in chlorophytes. That's found in the chloroplast genome. In charophytes and land plants, that gene is actually in the nuclear genome. And so this is obviously a molecular characteristic that was hard to explain when we thought that charophytes and chlorophytes were a monophyletic group. They're not. They're paraphyletic as we now understand it, and then it makes all of these characteristics, all these synapomorphies really make a lot more sense. Okay, the other thing that's shown on this slide is a bunch of synapomorphies for land plants. And I'm going to go through all of these synapomorphies sort of one by one as we go on. But before we do that, I want to come to sort of one of the main things that I'll emphasize throughout this lecture, and that's the life cycle evolution that we see in plants. And so plants actually have a different life cycle than the life cycles that we are used to thinking about. And this is a life cycle that we call the alternation of generations. And a couple of slides ago, I had, just scroll up here really quick, I had some different diploid and haploid, so having one or two sets of chromosomes, life cycle sort of highlighted here. And this evolution or change in the life cycles is really highlighted by this. And so I just wanted to point that out. I didn't talk about those here, but we'll come back to this idea of how the life cycle has changed in this transition to land, and then even within the land plant groups as well. Okay, so but first we need to understand what the alternation of generations is. And so you'll see this oftentimes drawn like this. I will show you multiple different types of life cycles to try and drive this home a little bit. But I'll start from a point that most of us are familiar with in terms of life cycles of other organisms or other eukaryotes. And that is with the formation of the zygote. And so in the life cycle of plants alternate between a haploid generation, which will often be depicted as 1N or N, and a diploid generation, which will often be depicted as 2N. And so this has to do with the number of chromosomes. We're diploid organisms. We have two sets of all of our chromosomes. Plants alternate between a haploid part of their life cycle and a diploid part of their life cycle. Already, this is a strange thing, and I'll give you some analogies to try and make this more clear of how this is different. But I'll start where we're familiar. We're familiar with gametes, sperm and egg. And we're familiar with how those will fuse. So sperm and egg are both part of the haploid generation. So these are both haploid. They have one set of chromosomes. And when those fuse, this is actually the process that we call fertilization. And fertilization is a transition from a haploid stage of the life cycle in plants to the diploid stage of the life cycle in plants. And so the first cell of the diploid stage of the life cycle is the zygote. And this is where the sperm and egg have fused together to form that zygote. This diploid stage of the life cycle is also called the sporophyte stage of the life cycle, or the spore-bearing stage. And as we mentioned, this is the diploid part of the life cycle. From that first cell, that zygote that represents the first cell of the sporophyte generation, we undergo mitosis. And so this is standard cell division. Mitosis occurs, and this grows into a mature sporophyte. The mature sporophyte, you can think of that oak tree outside the window, or that pine tree, or that fern. This is the mature sporophyte in most plants. We'll see how this varies in different groups of organisms, but for the purposes of this right now, that's what we're thinking about. So this mature sporophyte diploid organism undergoes meiosis in some cells, and that meiosis then produces spores. And so meiosis is our transition, is our process that transitions us between the diploid and haploid stage of the generation. And so spores are actually the first cell of that gametophyte generation. And so gametophyte is what we call the haploid generation. This is the gamete-bearing generation. And this is haploid. So spores are our first cells of the gametophyte generation. And so this is a little bit different, because what gets produced in an animal system, or most animal systems like ourselves that we're familiar with, meiosis actually results in the formation of gametes. That's not the case here. Here, meiosis actually results in the formation of spores. Those spores then go out into the world, they germinate, and they undergo mitosis, and they grow into a multicellular gametophyte. And so this is a separate part of the life cycle, where those, the gamete-bearing part of the life cycle is this multicellular separate organism that actually then produces gametes by mitosis. And so mitosis occurs to produce our gametes, our sperm and our egg. And then those fuse together and fertilization happens, and we move to the zygote, move through this alternation of generations. And so in plants, we're actually alternating between a haploid part of the life cycle and a diploid part of the life cycle. Now, as we will see, this varies in terms of which part of the life cycle is dominant throughout the life of different groups of plants, and which part of the life cycle might be independent, free living, or might be dependent on the other life cycle, not free living, dependent on the other stage of the life cycle for nutrition and growth. And so that those things vary. But all plants, all land plants actually undergo this alternation of generations in their life cycles. Okay, I'm gonna belabor this a bunch, and so we're gonna go through different ways of looking at this, but alternation of generations is weird. One, we're most familiar, of course, with our basic animal life cycle. The basic animal life cycle, I don't need to go too much over it, but we, all start out as zygotes. Those zygotes are formed by fertilization. Another word for fertilization is syngamy. And that was from sperm and egg that fuse together. So the syngamy occurs, and we form that zygote. That zygote then grows via mitosis into a mature adult, a mature organism. And then meiosis occurs, and meiosis occurs to produce gametes. And so that is sort of what we think of as that's our basic diploid dominant life cycle, where the only haploid cells that we have are our gametes, and those haploid cells fuse together and then form that new individual, and this life cycle repeats. That's sort of our view of life cycles in terms of, with that basic animal life cycle as our foundation. When we start to look at how this varies in green plant lineages, we see we see various things. When we look at the Charophycean green algae, so remember charophytes are that green algal group that's sister to all of our land plant groups, we see something a bit different. And so it's similar to that animal life cycle, but different in that these are haploid dominant life cycles in the Charophycean green algae. And so I'll go ahead and start again at fertilization or syngamy. That's the formation of a zygote from the fusion of sperm and egg, and and that zygote then immediately, so if we notice, zygote is the only diploid cell, in this is the only diploid cell in the life cycle is the zygote that immediately undergoes meiosis to produce a spore. That spore then undergoes mitosis to produce a mature haploid body, where those then undergo mitosis to produce our gametes, the sperm and the egg. And then fertilization occurs, those fuse, and we're back to that zygote. And so this has this haploid dominant life cycle where the zygote is the only diploid part. That zygote can hang out for a while, but when it's time to grow into another individual, we immediately undergo meiosis, produce spores. Those spores germinate and form through mitosis the main plant body. And so this is definitely sort of flipped on its head, right? Where we have haploid dominant part of that life cycle. I'll go back to the land plant, the basic land plant life cycle and that alternation of generations, where now rather than the diploid part of the life cycle only being the zygote, we actually have two separate parts of the life cycle. The sporophyte part of the life cycle, that's our diploid stage that produces spores, and the gamete part of the life cycle, which is our haploid stage that produces gametes. And I want to emphasize here that really we have this transition, sorry, I drew that a little bit wonky. It's the transition that are important. And so we have this transition from diploid to haploid, and that's meiosis that produces our spores. And then we have another transition from haploid to diploid, which is that fertilization. And so these are two separate independent parts of the life cycle. And so the way that I like to think about this is, if in our bodies, like if we just think back to humans, the gametes that I produce are sperm. And think about this as these sperm are put out into the environment, they actually multiply into a multicellular organism that then produces single cells themselves, that the gametes, their own gametes, that then fuse with an egg cell to produce a zygote. And so it's like if our gametes were to go out and produce their own separate little organisms, whether those are sperm or egg, and then those undergo mitosis to create the gametes themselves. So it's these separate little organisms whose sole purpose is to get out in the world and to make gametes. This is a really different way to think about it. I'm glad that our gametes don't do this, but if we think about it like that, it sort of puts this into perspective. Like, we have these separate organisms that can actually be independent organisms producing gametes that go out and fuse to produce the zygotes and grow into the mature diploid organisms that we see in the environment. So pretty wild, pretty different, and a little bit mind-blowing. And so, and unique to land plants. And so all land plants share this alternation of generation lifestyle in terms of their life cycle. Okay, so if we then look back at our sort of group of synapomorphies, recognizing that green algae were paraphyletic, we're able to reconcile a lot of those characteristics that are leading to the rest of the Virida plantae, so Charophycean green algae plus the land plants. We then see that we have a bunch of synapomorphies for land plants themselves. There's one on here. The one that's not listed on here that I want to put right now is the alternation of generations. So there are 6 synapomorphies. Alternation of generations. There are 6 synapomorphies for land plants, and so alternation of generations is one of them. So that life cycle evolution is an important one. The second one, kind of an obvious one, is transition to land. So we have land as part of the alternation of generations. A third is the embryo, and we'll talk about this, having multicellular reproductive structures. And so this has to do with how those gametes are actually formed in the gametophyte. So the gametophyte has multicellular reproductive structures. The presence of a cuticle, I mentioned that briefly last time. We'll go into detail about what that is. And then finally, having spores that are trilete. And I'll define what that is. That's because of meiosis producing those spores. They end up with a scar on them that we can see, and I'll show you some good pictures of that. So we actually have 6 synapomorphies for land plants that are shared by both non-vascular and vascular plants. And so what I'll do now is actually start to look at those individual synapomorphies for land plants and how they, and what those look like. The first of those land plant synapomorphies is the cuticle. And so here we see a cross section of a leaf. So this leaf cross section, looking at the different cell layers, and the important part of this that we're looking at here is actually this waxy layer that's found on the outside of the plant. So this is a waxy secretion from the epidermal cells, whose main function is to deal with one of the biggest problems of moving to land. And one of those big problems is preventing desiccation. When you're in an aquatic environment, you don't have to worry about desiccation, but when you are all of a sudden exposed, desiccation is a big problem. All those water-filled cells are going to lose it, but this cuticle actually prevents that. And so the main function of this, of the cuticle is to prevent water loss. Okay, so that's our first land plant synapomorphy, is the presence of a cuticle. So all land plants have this cuticle, this waxy layer on the outside of their body. Okay, the second one of these is having multicellular, the second synapomorphy for land plants is having multicellular reproductive structures. And so those multicellular reproductive structures are found in the gametophyte generation, since this is where those gametes are made. We see that in the gametophyte generation. And what I have pictured here are 2 different multicellular reproductive structures, one for the male side and one for the female side. And so on the male side, we have sperm that are produced inside of what we call antheridia. And so this is an antheridium that's depicted here. And this is that multicellular reproductive structure for the male. All land plants have this. In a couple of groups, it's been reduced, but it's clear that this came from this multicellular antheridia. On the female side of this, we have eggs produced inside of archegonia. And so archegonia is our multicellular reproductive structure on the female side of this. And so what we see here is an egg cell that's produced within this individual archegonium. And in some groups, sperm will actually swim. And yes, some plants do have motile sperm, will actually swim down that neck part of the archegonium and enter where fertilization will occur and form that, and form fertilization between the sperm and egg will occur and form a zygote inside of that archegonium. So the archegonia are the female reproductive structures, and the antheridia are the male reproductive structures. And we'll look a bit more at some of these in specific groups. Okay, so that's two of our synapomorphies. Our third synapomorphy that I want to tell you about is having trilete spores. And so trilete spores refer to this scar that we see on every spore. And why is this three-sided scar seen on these spores? And that's because, remember, spores are the product of meiosis. Remember, spores are the first cell of the gametophyte generation. Spores are the first cell first cell of the gametophyte generation that germinate and form that mature gametophyte. But they are the product of meiosis. And remember, meiosis has where one cell divides into 4 daughter cells. Those 4 daughter cells are initially stuck together. When they separate, each one of them shows the scar of where the 3 other daughter cells were attached. And so we see these trilete spores. This is an important one because this is one of the first evidences of land plants is because in the fossil record 450 million years ago is the presence of these trilete spores. These spores are covered with a compound called sporopollenin. Sporopollenin, move this over here. Sporopollenin is a complex polymer that does a couple of things for these spores. It allows for, one, desiccation resistance. Remember, we're moving on to land. This dealing with desiccation is a big deal. Cuticle also dealt with desiccation resistance. Well, this is dealing with desiccation resistance in the spore stage of the life cycle. The second one of these also deals with an issue that we have on land, and that's UV radiation. And so this is protection against UV radiation. In water, in that aquatic environment, UV radiation, especially in deeper systems, is not as important as it is once you move on to land and have to deal with that part of that, another challenge to moving on land. Okay, so having trilete spores with sporopollenin is another of our land plant synapomorphies. That's 3. I think our 4th land plant synapomorphy is the young sporophyte. So once that sperm and egg fuse together, they produce the zygote. The zygote begins to divide by mitosis and produce a young sporophyte. That young sporophyte, which we call an embryo, just like we do in other systems, is supported by the gametophyte generation. And so if we look at this moss sporophyte developing, so this is in a moss, we see here the embryo inside of the capsule. This is the embryo. And so that's the sporophyte, sporophyte. And so that's diploid. That's that young sporophyte developing. And it's actually supported by the gametophyte. And so all of this tissue up here is actually gametophyte tissue. And you might notice another familiar word on here, and that's the placenta. And so the placenta actually is a place that transfers, or tissue type that transfers nutrients from that gametophyte to the sporophyte, to that developing embryo. And so this is a synapomorphy for all land plants, where we have the young sporophyte, the embryo, actually being supported by, retained on the gametophyte and supported by that gametophyte through this placental transfer. That is an important thing, and actually is where the name, the alternate name for land plants actually comes from. So we call, if you recall, we call land plants embryophytes. We call them embryophytes because the embryo is one of the synapomorphies, the embryo being supported by that gametophyte. That's why we call these embryophytes. All right, I'm going to end this video here, and then when we come back, we'll dive a little bit deeper and start to dissect out some of those other land plant lineages and the different adaptations that they show.